By the way, I'm not Dr. Feelgood, and I don't play him on TV either. I'll tell you why I came up with the name for this, or how I came up with the name of this talk. But my name's Dr. Goff. I'm a chemistry instructor at Western Wyoming. I've been here for 4 years. This will be my 5th year. What we're going to talk about today is drug discovery, okay? And I'm not talking about finding out like you got an extra pill of antibiotics you got to take. What we're talking about is how do drugs actually make it through the process of being approved by the FDA and get to market. So I'm going to talk a bit about the FDA, background on it, what its purpose is, some of the important steps that you would have to go through if you were a chemist or you were someone involved in drug discovery. And then what we're actually going to do is we're going to go on a little journey together, and we're going to talk about a specific drug called Colloquium. And we're going to discuss about how it was discovered, how it went through the FDA process. We'll talk about some of the work I did on this particular drug, and then some of the pitfalls that were found, and then actually some of the positives that came out from this drug being able to get to market. So, how did I come up with the name for this talk? Well, last year we had a faculty member, Dr. Sandy Mitchell. Unfortunately, she passed away last spring, but she kind of set the standards for the faculty members to come up with some really gnarly titles for our talks. Hers was called Quickies in the Desert. I won't go into the details on that, but it is like what you're thinking it is. And so I had to come up with a really good talk title. So I always liked Motley Crue when I was younger, and one of my favorite songs is Dr. Feelgood. And then I really like Stanley Kubrick films. He was the guy who did 2001. And he had a really nice comedy, one of the few comedies he did, called Dr. Strangelove, or How I Learned to Stop Worrying and Love the Bomb. So it was a black comedy about nuclear war. And he actually made a pretty good comedy about nuclear warfare, believe it or not. So I came up with a title based off of that. So just so you have a little background about me, I was an undergrad just like you. I went to a junior college. I majored in chemistry. I ended up going to Long Beach State. I'm a Southern California native. I started getting my feet wet with doing actual chemistry in the lab. I did some research with a faculty member there. I went on, I got my PhD at BYU in the chemistry and biochemistry department. And my background is medicinal chemistry, which is essentially making molecules that can be used in a pharmaceutical sense. Okay? When I finished with my PhD, I went on to the University of Wisconsin in Madison. And I got to be honest, when you guys talk about UW, I'm thinking about this. Okay? I'm not thinking about the brown and the gold one. So I was a postdoc in the pharmacy school there. I did a lot of anti-cancer research. And the research that I'm talking about today is based off of what I did at Wisconsin. And then I went on to University of West Florida. I had my own research lab. I worked with undergraduate students there and kind of continued some of the same work I did at Madison. Okay. So let's talk a little bit about the FDA. I know there can be an attitude of skepticism about how well does government help us in some aspects. And I know that right now, especially in the climate we might have, there's really a lot of issues about, is government maybe interfering in things? Things like big corporations, are they really driving, kind of what goes on? And really what I want to give to you is an optimistic view about the benefits that we actually get from an important governmental body, and that's the Food and Drug Administration. Okay? So just a little bit of background. It actually started in the 1800s as a part of the U.S. trademark, U.S. Patent Office. So at this time, there were vendors, and they started selling things, saying like, hey, you could take this and your hair will grow back, or take this and you'll lose weight, or take this and this can happen. And it kind of sounds like a lot of late night TV ads, right? Well, actually the government came out and says, well, wait a minute, we've got all these products on the market, and are they really doing what they claim they're doing? Are the ingredients really what they say they are in these products? And so the government said, look, we need some sort of a chemical analysis lab that actually verifies what these compounds are supposed to be doing. And so the U.S. Patent and Trademark Office had their first office of chemical analysis that would do that. Then when the Department of Agriculture came along, they actually spun that part off into a whole different division, until finally in the 1900s, where there wasn't a whole lot of teeth given to what was then the precursor of the FDA to really enforce these practices about, hey, if you're going to sell this, it better do what you claim it does. So finally, about the turn of the century, right about the time when there was a lot of social changes about looking out for the consumer. The Congress passed the Pure Foods and Drugs Act, which gave the FDA a lot more teeth. And then finally, right in the 1930s, that's when really kind of the modern day FDA came to be what it is. There was more legislation that came down that gave the FDA even more regulatory powers, along with the FTC, where now when we have something on the market, it better do what it says it does. Okay? And really the Food and Drug Administration is, I would have to say, one of the biggest advocates for our health and safety. I mean, I pulled this straight off the website. They're looking for the safety, efficacy, and security of the devices we use on the animals that we use as pets and use for food sources and things like that, to make sure that what we're handling are safe and effective. Okay. So one of the things that you might think about the FDA is they're just really out there just to deny. We don't want you to have this. We don't want you to take that. And yeah, there is a certain an element to that, where really the FDA is there just to say, if there's a drug or a compound, something that you're going to take, we want it to do what it says it's going to do, but we also want it to be safe. Okay. So some of the things that I kind of pulled from my my memory and some of the things that I learned going through school is, there were a lot of compounds that have been on the market. I've got a few here. There's a really big laundry list of other compounds, other drugs, that the FDA says, these have no medicinal value that we know of, or they do, but they cause some pretty nasty side effects. One of the ones I personally remember was fen-phen in the 90s, and this is a really fad drug. It was a combination of two drugs. If you took them in a certain proportion, it would help you to lose weight. Problem was, years later, as they found out that people were starting to have problems with their heart, their heart valves were failing, and people were passing away because they had been taking fen-phen and needed all kinds of operations. And so, fortunately, we have that level of care where we have scientists that are actually looking into, are these compounds safe, or are they going to cause some problems that we need to pull them off the market. Okay? But the FDA isn't just about banning things, they're also about promoting health. One of the big things I can think of happened in the late 80s, where the FDA said, we need to develop packaging that can't be tampered with, so that you would buy something and take it, and there's something that would adulter what you have in that package. Does anyone know what precipitated this? Probably some of the older folks. Something happened in the early 80s. You know, Rocky?
I'm paying attention to how many of these students are screwing around on their phones, not listening to a single word that you're saying. I know that most of you are in here for the FYE as a requirement. So if you can't put your phone away, you can't stop looking at Facebook, get the hell out. Now, what was your question, Dr.
Okay. Does anyone remember what caused this to happen? There was a big event that happened in the 80s. I'm sure most of you, you weren't alive at that time, but there was something that happened that people were dying because they took something that had been messed with in the stores. Does anyone happen to know? So back in the late 80s,
Tylenol.
Yeah, it was Tylenol. So it turned out in Chicago area, someone had been going in and opening up packages of Tylenol and were putting in cyanide, and people were taking this and they were dying left to right, and it took a while to find out what caused it. And to this day, no one really had been caught for tampering with the Tylenol. So you could literally go in, get a bottle, open it up, put some stuff in it, put it back on the shelf, and take it, and there you go. So that's one of the things that the FDA implemented that has helped to give us some safety and security with the drugs that we take. Another thing I could think of is 1999, they established a website called clinicaltrials.gov. So if you're an informed patient, and let's say you've been diagnosed with a disease or a rare syndrome, and there's really no cure for that, you can go right on this website and you can look to see who is doing a study on this disease or using this drug to cure this disease. And an informed patient can actually go to their doctor and request information to be put on a particular clinical trial. So they've actually made it easier for us as potential patients or consumers to be more aware of what kind of research is going on that, as someone that might be dying of a particular disease, maybe I want to be part of, that would help cure me, or if not, maybe extend the knowledge we have of science. Okay. All right, now the biggest one that I can think of, and I'm not going to spend a whole lot of time on this one because I want to get into some other stuff. The biggest impact I could think, single-handedly we can point at and say like, wow, the FDA really came through on this one, was a drug called thalidomide, okay? And I'm sure most of you probably don't, this is even before my time, but thalidomide was a drug. It was discovered in the 50s by a European company. They thought, man, this would be a great drug to cure insomnia or perhaps a good cold medicine. But even better, they says, hey man, this stuff is going to be great if you're a pregnant woman and you have problems with morning sickness. This would be good as an anti-nausea medication. So this was marketed in Europe. They came over to the U.S., the Swiss company, and they tried to get U.S. companies to buy in to the thalidomide drug, because to use it in the U.S. at this time, you have to go through the FDA to get their approval to put it on the market. So what the FDA essentially did is they went back to this company and said, we're not going to put this on the market. Your data is inconclusive. We're not sure how safe it's going to be, okay? Did anyone ever heard the story about thalidomide? Okay, Eric, do you know what happened as a result of that?
In Europe, a bunch of pregnant women took that drug and they had massive birth defects.
Yeah. Really horrible birth defects from their offspring. Typically what happened is their limbs were stunted, or their hands, their feet were kind of like flipper, like a seal. They just were malformed. Some of them had some malformation of their head, their brains, and about half of the infants that were born died fairly quickly. And the reason why we didn't have this issue in the U.S. is because the FDA, someone at the FDA was brave enough to stand up and say, we don't know enough about this drug to make it safe for the consumer to use. So this particular physician, Dr. Kelsey, she actually won, or she was awarded, I can't remember the award, but one of the highest that you can get from the government as a civilian. She was recognized by President Kennedy just because that, she, didn't bow to the pressures of a company coming in and saying, here's a load of money, we want to get this on the market. So the U.S., we had very little birth defects because it was only very few people actually went over to Europe to get the drug and use it, and that's what ended up happening. Okay, so sometimes as a chemist and working in the drug field, so you're working for a big pharmaceutical company like Pfizer, or a little tiny biotech company that just got started maybe a few years past, sometimes you feel like this guy, Sisyphus. So this was a Greek mythological figure, and he was punished for doing something wrong, and he was supposed to push this rock uphill for all eternity, and he would get it up the hill, and the rock would roll right back down, and he would start pushing up the hill again. And sometimes in my field, it could seem like that because it just takes so much effort to get a drug to market. So what does it actually take to get through the whole FDA process? Okay. Well, if you're a large pharmaceutical company like let's say like Pfizer or Merck or Novartis, there's something that you're going to research. Okay. It might be an antibiotic. It might be something to do with curing cancer. It might be an antifungal. Maybe it has something to do with HIV or a viral thing. And essentially what you do is you design a lot of different drugs. So you actually go in the computer, you do some modeling, you design a whole slew of different drugs, and you might have about 9,000 to 10,000 candidates, okay? Then it takes a long process of many years of whittling through all of these candidates to find out what's going to work and what's going to be safe. So with just in a few years, you go into the lab, the chemist makes the drug or makes these groups of drugs, then they test them, okay? So what's the main thing you want your drug to be able to do? You want it to do what it's supposed to do. So if it's going to cure cancer, it better do something with killing cancer, okay? If it's going to grow hair, and it hasn't happened yet, it better grow some hair, all right? So you have to make sure it's going to work, but then you have to make sure, is it going to start killing what you're going to put it into? So before you even get it into people, what do you think would be a smart thing to do? Test it, but test it on what? Mice, dogs, pigs, okay? All right? You want to know, is it going to kill, is it going to harm your liver? Is it going to harm your heart? Is it going to harm your brain? So there's different animal models that you would use to find out how well is this drug going to, well, is this drug going to cause any side effects? And so there's different animal models we'll use that will mimic different organs that people have. So for example, I remember when I was at Wisconsin, one of the models we would use was beagles, because for some reason the blood-brain barrier, from what I'm told, of beagles is consistent with that of people. And if you could look at the neurotoxicity or how well a drug would kill your brain, if it didn't kill a dog's brain, it probably was okay for people, or at least it would be a decent model for people. So you would go through this whole long ordeal, which is necessary, of looking at, you know, how well does it work in animals, and then looking at people. Having something in a dish, does it really work well in the lab is one thing. How well does it work inside of a person is a whole other thing. So going through the FDA process, you go through a number of what are called clinical trials, and each clinical trial becomes larger and larger than the next. And the criteria gets set up by doctors that you have to hire. The doctors have to have staff to draw the blood. They have to have people in the lab that do the analysis. So all of this isn't one person's undertaking. It's a whole group of people that are people from chemists, microbiologists, biochemists, molecular biologists, doctors, nurses, okay, lab workers. It's a whole collaborative effect. And by the time you get all the way through the FDA trials and you find out, you know, the drug works okay, it's not killing people, it's curing people decently, you get to the FDA and you still have to pass the application process, the review process. Finally, 16 years later, boom, you got one drug out of 10,000. Okay? So what do you think about the cost of drugs? You know, sometimes we think, wow, it's really high. It's no fair. One round of treatment costs $10,000. But when you see the whole work that has to go into making one drug, then you start to get an appreciation of why drugs can cost so much. Not going into the ethics of why, you know, they need to be high, but it really explains why drugs cost so much. Okay? So sometimes medicinal chemists really do feel like this. I've known people who've gotten all the way down here to the last step. Drug didn't pass FDA review, 15 years, kind of almost down the tube, sort of, a lot of money, you know, spent. Okay? Now, let's talk about an individual drug, really interesting compound. Okay? So I realize none of you guys are chemistry majors. I'm going to kind of assume a few things in here. Namely that, you know, well, I'm not going to worry about you guys knowing exactly what I'm showing you in some of these graphics here. I'm just putting them up just so you can get an appreciation that there are a large amount of chemotherapeutic agents. So in other words, compounds that would kill cancer cells. Okay? And what I want you to notice is each one of these looks different, and each one of these actually affects DNA in a different way, or affects cancer in a different way. Some of them bind to DNA and keep it doing from what it needs to do. Some things keep DNA from replicating itself, or even keep cells from dividing. And then the compound I'm going to deal with today is called colloquium. And I've got two words for you. Okay? Ready? Okay? Fruit Ninja. Okay? All right. If you know the app, you know what I'm talking about. I'll talk about that in just a second. But colloquium is interesting. Okay? It's a big molecule. There's three really cool parts about it. There's a neat little ring right here that has a lot of electrons in it. And these electrons can form interesting new bonds when it reacts. Okay? There's another part here. The S's are sulfur atoms, and there's three of them in a row. That's really rare. We never really see that in a compound. The even coolest part, and this is the part that makes me so excited. Oh, you guys, you can't even appreciate how excited I am when I see this. And that's definitely a very nerdy confession on my part. Is this part right here. I'm a carbohydrate chemist. I've studied sugar molecules and how they affect drugs. And we've got a sugar molecule here, one here, and right in between there's an oxygen-nitrogen bond, and that's rare. You never see that anywhere. You usually see sugars bonded to an oxygen atom. And I'm going to talk about why this is so exciting. Okay, so colloquium. Really interesting story, okay? So can you imagine going on vacation and halfway through your vacation, you kind of get into your work, you get into your job? Can you imagine being so into your work, your job, that you're in the middle of your vacation and you're like, you find something that would be so exciting that you sort of interrupt your vacation? Can you guys appreciate that? Okay, by your excitement level, then yes, I could see you appreciate that. Okay, well, there was a microbiologist in the early 80s, and he's living out in Massachusetts. He works for a pharmaceutical company, and he goes out to Texas on vacation, and he's an amateur rock hound, okay? So amateur geologist. So he finds this interesting looking rock, and it's called a caliche rock. It's limestone based, and it looks different from all the other rocks in this area where he's hiking, wherever he is in Texas. And he says, wow, this is really interesting. I'm going to box this up, and I'm going to send this back to my research lab, and we're going to look at this rock and see if there's something interesting that it contains, okay? So what do you think about that right there? Can you just imagine doing that, like you're out having fun, like you're in the middle of Disneyland, and you're like, wow, that's really interesting gum stuck on the ground. I'm going to peel that up, and I'm going to send it, right? I mean, that's not really something you think about, but really, one of the big ways we make scientific discoveries is when sometimes we're not even looking for it, it just kind of slaps you in the face. And that's really how penicillin was discovered. But Louis Pasteur, he was the French scientist who was really the father of microbiology and developing vaccination. He says that really is no luck, it's just chance. Chance favors only the prepared mind. So if you're ready to see something, you're going to recognize it for what it is. So what this microbiologist did was he cultures it or tries to find bacteria that is actually growing in the rock. And he actually identifies one or several species of a particular genus of bacteria. And he runs an assay on it. And this assay is just to find kind of what biochemical reactions, do these bacteria do, okay? And he finds really one interesting reaction, and that's killing DNA. It's something's killing DNA, okay? And it takes him 2 years to go through this whole screening process. So can you imagine this? He's found this rock, and he's still 2 years later, he's still studying this rock to really understand what's going on with the bacteria in it. And he identifies that only 1% of all of the compounds that he's discovered that this bacteria is making, only 1% is killing DNA. So at this point, he's like, I've got to know what this is. So they do more work, and they turn to the chemists at his company, And a few years later, they discover what the structure is. And the structure I showed you on the last slide is this compound colloquium. Okay? Well, they published the structure in a scientific journal, and another chemist by the name of K.C. Nicolau, in the chemistry circles, he's a really, really big name. This fellow works at Scripps Research Institute in San Diego. He builds the molecule from scratch. So takes teeny tiny molecules and puts them together, makes this very, very big complex molecule. Sort of like, why do you climb a mountain, right? Why do you climb Mount Everest? To get to the top, right? Because it's there. Well, that's why he makes this, is because why not? We want to make it. And another a scientist by the name of Peter Townsend at Johns Hopkins actually figures out what this compound does. So in the space of about 16 years, from the time we discover the rock, we finally figure out what does this compound do. So what were the two words I said to remember? Fruit ninja. Okay, let's see how that works. Okay, so here's colloquium. I'm just showing you the business end of colloquium. Okay, so this gets entered into a cell brings this inside. Okay, and something in the cell, it could be anything, something that's reactive, will add on right here. And this chemical bond breaks, and we make something called a thiol. So this little red part here, okay? And this thiol is reactive. And there's another part that's complementary on the molecule right there, and it reaches around and it grabs onto that molecule, okay? And when it does that, it takes this little ring section, and this ring section starts to get pushed closer and closer together. So there's these two little arms start to get squeezed. And when they squeeze, something really cool happens. The electrons in these chemical bonds start to shift location, and they cyclize, making a new ring. And these dots here represent radicals. And you might have heard the term radical before. A radical is something that can cause damage to your cells. So if you've ever heard, in nutrition circles about taking antioxidants, these are substances that will scavenge radicals. Radicals left by themselves in your cells can cause damage and cause cell death. So we take antioxidants to eat up things like this. Well, in the case of colloquium, it forms this diradical. And when it gets close to DNA, it takes parts of the DNA, it takes some atoms away from the DNA, and the DNA now becomes radicalized. And when it's radicalized in the presence of oxygen, it cuts itself in half. So what's the fruit ninja app? What's the whole object of fruit ninja? Right, you slice the fruit up, right? Right, watermelon, right? Well, that's what colloquium is doing. It's the fruit ninja, it's the drug ninja. It comes in and it slices DNA up into pieces. Why do you think that might damage DNA if you cut it to a bunch of little parts? What does DNA do in the cell? It's genetic material, right? And it's used to make things like proteins and enzymes and signaling things. And if your DNA doesn't work, it's sort of like your brain's not working and you die. This is, in a way, sort of like the brains of the cell. If it's not working, the cell's gonna die. So in this period of time, we've discovered this drug that can kill DNA. And if it kills the DNA of cells, couldn't it kill the DNA of cancer cells? And if the cancer cells die, right, then you get rid of cancer in a person. So if you can find a way of killing cancer cells by killing the cancer cell's DNA, couldn't you conceivably cure someone of cancer? Does that sound exciting? Okay, exciting enough for a Monday afternoon. Okay, good enough, all right? Well, interesting enough, a chemist at Cal Berkeley actually discovered this same thing before the drug was even found. And he discovered that if you have a structure like this, and that's the structure we see in colloquium, if you heat it up enough, those two little arms will come together and they'll cyclize and you'll get a diradical, and that diradical can do some really gnarly chemistry. Okay? So even before this drug was discovered, nature had it in its little back pocket of a little technique to use to kill things, to kill cells. Okay? I think that's really, really slick. okay? And this is an actual model of what colloquium, not a model, sorry, it's actually a little snapshot of what colloquium actually looks like with DNA. So you guys have all heard of MRIs, right? Okay, what's MRI do? Yeah, it scans you, right? You go in that tube and it's a big magnet, and when it scans you, people can look at, what's your brain look like, your heart. Yeah, like you get a concussion, what does your brain look like? It tells you more information than an x-ray, right? Well, this is an MRI for molecules, okay? And someone actually got a picture of this. So here's DNA. The purple parts are the kind of the greasy parts of DNA, and then the yellow parts are the backbone. Can you see the double helix? Right? There's one, there's two. And here's colloquium, and it fits right in DNA. And there's the business end, there's the fruit ninja part, and it fits right there. And when that turns into a circle, it cyclizes, you make the diradical, and that's going to react with DNA. So can you see how close that is? It fits right in there, and it can react with DNA. Okay, you feeling the goosebumps already? Yeah? Okay, I am. All right, good. Okay, so how good does this work? How well does this work? Well, colloquium is like the wonder drug. Man, if I could make every drug like this and every anti-cancer drug, wow, that'd be awesome, okay? When chemists look for drugs that work well, we want the drugs to work as best as they can with little amount as they can, because the more drug you're taking in, the more foreign substance you're taking in, right? And if you're taking in, what was that?
Could cause side effects.
Because it could cause side effects. So the less you're taking in, you can minimize the side effects, right? So what we find with colloquium is numbers that are, and I realize that unless you're into chemistry, this isn't going to make a whole lot of sense right now, but they're on the nanomolar range. And what we mean by nanomolar is we're talking about if you have 1 billion particles, one of those particles would be like a nanomolar quantity, okay? So we're talking about if you can have 1 particle per billion that does the job, that's not a whole lot of material you have to be ingesting, okay? So that's actually a good thing, a positive thing. So the lower this number, the better the drug works, the stronger the drug is. And you could see by all of these types of cancer drugs, cancer lines, so breast cancer, ovarian cancer, prostate cancer, it's very, very very low numbers. So we don't need a whole lot of colloquium to do the job. And B cell leukemia is even better. That's a tiny, tiny number. We're talking down to the parts per trillion. So just a minute amount works awesome against B cell leukemia, okay? So doesn't this sound like, hey man, it's a wonder drug. We got the cure for cancer right here. I mean, doesn't that kind of like make you think like, well, hey, cure for cancer. I mean, what do you guys think? Yeah?
Yeah.
Yeah, okay.
It doesn't infect your body cell.
Yeah, but what's one of the problems though?
Great.
Yeah, it's going to kill everything, right?
Yeah.
It's kind of indiscriminate, okay? It's not just going to kill the stuff you want to die, it's going to kill your healthy cells too. And that's the big problem. So you have to have something that works, but something that's only going to work on what you want it to work on. And healthy cells, yeah, it's going to kill those too. So that's a big problem. It's going to wipe everyone out. So that's not a viable alternative, okay? So what do? We just take the research and say, let's take all those millions of dollars and just, okay, we'll just write it off and we'll get a tax, write off from the IRS. No, you don't do that. You kind of innovate. You think about creative ways of, could I make this actually better? How can I make it better? What can I do? So we're thinking of little tricks here, okay? Well, early in the 70s, scientists came up with this idea of cloning something called antibodies, right? How many of you guys have heard of antibodies? Okay, antibodies are part of your immune system. They're sticky. They stick to certain things that we call antigens. So if you've got a cell that recognizes a cold virus, it can stick to that cold virus and say, hey, got a cold virus right here. Body, come in and kill the cold virus. Okay, we got a foreign invader. So these antibodies are really important as little things that you can use to identify stuff that you don't want to have in your body, okay? All right, thanks for that. So what scientists did was they took mice they would expose them to something like a cold germ, or they would implant cancer in them, or something that you wouldn't want to have, okay? And what they did was they collected the spleen cells. So the spleen cells are part of your immune system that are involved with, killing or identifying foreign cells. And they took those cells and they fused them with a cancer cell, okay? Now, what does a cancer cell do? Eats, multiplies, right? Eats and multiplies, That's all a cancer cell does, is eats and multiplies. So they took the eating and multiplying part of the cancer cell and fused that with the operation of a normal cell that identifies these foreign compounds, and they essentially made a cell that can live forever. So we made something called an immortalized cell or a hybridoma. And these are cells that essentially, as long as you give them enough food, they can just about live forever, okay? So you can keep cultivating and cultivating and cultivating. And what we can do is we can select for certain antigens. So, hey, you want the antigen for the common cold? Boom, we got a cell for that. You want an antigen for some, something that's going to cause, something bad to happen? Boom, we got the antigen for that. So then we can collect these antigens, and these antigens act like little magic bullets, right? These are going to stick to the parts of the foreign cells or the disease cells you want to get rid of. So it's a really, really convenient way of targeting a drug right to that position, okay? So how do you think this could be useful for colloquium? If colloquium is killing everything, how do you think something like a directed attack here can help?
To target the healthy ones for resistance?
Okay, you could target a specific cell. So maybe you want to target the healthy cell for some reason, or maybe you want to target a disease cell, okay? And you can use that to go after one specific cell and not kill everything, right? Doesn't that sound kind of like a spark bomb?
Yeah.
Right, bomb's just going to hit that one target. It's not, you're not going to have collateral damage, hopefully, okay? That's essentially what we're doing, is we're setting up a little smart bomb for colloquium. Well, monoclonal antibodies actually are approved clinically by the FDA in a lot of different types of cancer, and also some other, well, I don't know if asthma is really a disease, we'll call it disease. Some other negative issues that people can have. So essentially, what the idea is let's take an antibody. So here's the shape of an antibody. It kind of looks like a Y. It's a protein. And what you would do is you would take your drug, so colloquium is like the little green circle, and you put a linker on it, and you attach that to your antibody, okay? Then, when that gets in your body, it goes directly to the diseased cell, because it's only going to recognize the cell that has the counterpart that it can recognize. So it's only going to go to certain cells that it can recognize, and if it's only recognizing the disease cells, guess what it's going to kill? Just the disease cells, right? Okay, so this is what happens. So this company, this chemist or this microbiologist that found this material, his company's been bought out several times. It got bought out by Wyeth, which is now part of Pfizer, and they said, hey, remember B-cell? B-cell leukemia had a very, very specific or low dosage that you needed for colloquium. So let's look at B-cell leukemia, and acute myeloid leukemia is B-cell leukemia. So myeloids are cells that come from your bone marrow. Okay, is that right, Mr. Biologist? Mm-hmm. Okay, good, all right. And what this disease can do is it can essentially kill your bone marrow, which you need for producing certain immunology cells. Okay, it can kill production of your red blood cells, right? We need that for oxygen transport. And of all the deaths in the U.S., it's a small percentage, but if you're one of that 1%, I'm sure that's significant to you. And the cool thing is, there's a thing on myeloid cells, the CD33 antigen, which is just a fancy name for a specific signal that's displayed. So if you could get an antibody that would complement and just stick to that antigen, now you've got a smart bomb that can be focused just on those disease cells. And that's what exactly Wyeth had done, the company Wyeth. So here's their antibody, here's colloquium. They put a little linker on there. So this is little chemical molecule. And what would happen is this linker would dissolve or would break off, and then the drug would be free to do its thing. So this is the first drug to ever make it to the market. It's called Mylatard. So that's the fancy name, and this name here that I'm not even going to try to pronounce, that's the generic name, okay? And it was the first drug that ever got to market. It got approved by the FDA. The first drug that is connected to an antibody. And because, you know, the company or the people at the company, they were so worried that this might still be very, very toxic, they wanted to look at a very limited group of patients that were pretty much, this was their last resort. If this didn't work, they were probably going to pass away from leukemia. So they only looked at patients with this particular type of leukemia. This happened in the year 2000, and they only looked at patients that were over 60 who had the disease, had it treated, it went into remission, but then it came back. So they were in relapse, and it was a very, very small pool of patients, okay? So they wanted to look at this very small pool to see how well it worked, so they're being cautious, and really this is what we believe that happens, okay? So you've got your antibody here, and these little spots are your drug. Okay, it's going to stick to the cell that has the specific antigen or the specific complement to it. Then it gets brought inside the cell, and once it's inside the cell, that whole thing is going to start to fall apart. Now your drug is free, and it's going to enter into the nucleus, and it's going to start doing what to DNA? What's going to do? Slice it up because it's the Fruit Ninja. Good. Okay, you're paying attention. And it's going to kill the cell, it's going to kill the DNA, and then the cell's going to die because the DNA is not functioning. Okay? So, what was the outcome? Well, it found out there was a higher mortality rate for this group of patients. So they took a group that got the drug and a group that didn't get the drug. And the group that got the drug actually had a higher mortality rate. So more of them were dying off with the drug. Okay? Why this happened? Well, they think like maybe the drug was coming off before it even got into the cell, and it was, you know, affecting other cells that you didn't want affected. Maybe that a lot of the drug got onto the antibody, so maybe only 2 or 3 molecules would get on, but maybe more were getting on. And the other problem is you have healthy cells that actually express this antigen. So maybe it wasn't just killing the bad cells, the disease cells, but it was killing the healthy cells. Okay, does anyone have the time?
12:45.
It's 12:45. Okay, I'm going to wrap this up really quickly. And the FDA, they came out and they said, hey, we got to take this off the market. We just don't know enough about what's going on that we want to make sure that people, you know, are going to be safe. Okay. Well, this is where my role comes in, and I'll try to be really brief. I worked on sugars, and I looked at how do sugars impact drugs. We know that sugars can affect how drugs work, okay? If you attach a sugar to a drug, it might get into the cell a lot easier. Certain sugars can make a drug work in a certain way, and if you change the sugar, the drug might work a little bit differently. So I looked at how can you put sugars on molecules really easily. And the bottom line is you can't. It's really, really hard. You have to go through a lot of little steps to get it on the drug. But we discovered in my lab at Wisconsin that if you modify the thing you want to put the sugar onto, you can get this to work sort of like a lock in a key, right? You stick if you have a key, it only fits into a certain lock. Well, chemically what we find is if we put this little oxygen-nitrogen tag on a molecule, sugars will just glom right onto it, just like a key fits right into a lock perfectly. These sugars glom right onto it. And the cool thing about this is you can make a big, diverse pool of compounds really, really fast. And you can test all these different compounds to see how well they work, okay? Well, I looked at colloquium, and we have been using this for another application in my research lab. And guess what? Maybe this is the exciting part I told you about. I got goosebumps on it. Right here is the oxygen-nitrogen bond that you never see in compounds. And that looks just like that little tag that we would put on a molecule. So I said, well, wait a minute. If we can get a sugar to pop on that why can't I get a sugar to pop right on that? And maybe there's another way we could tag colloquium besides using an antibody. So that's what I did. I went to the lab, and literally this took me 1 day. These experiments never take 1 day, but this one literally did. And I put a sugar on it, ribose, and that's the sugar you find in RNA, okay? And I put a little acid in it to make the pH low, and I put it in some solvent, and I reacted them, and I actually got the ribose sugar to pop right on there in one step, okay? And then I wanted to see how well does this drug work now, okay? So I modified the drug. Does it still work? Does it not work as well? Does it work better? And if these are the values for the native drugs, so the ones that actually are made by nature, and these are the ones that I made, so these are the involved my analogs, you find that the numbers are higher, okay? Meaning that they don't kill cancer, as many cancer cells at that lower amount. I have to have more drug to kill the same amount of cancer cells as the lower amounts, okay? Now that's still okay because the numbers are very, very low compared to many anti-cancer drugs, but still not as good as colloquium, okay? But could you potentially see an advantage for these numbers not being so low?
They don't kill healthy cells as quickly.
Yeah, maybe they don't kill healthy cells as quickly, so that could be an advantage then. So one of the things I'm envisioning is maybe we could put a tag, a different kind of tag on the sugar, then put the sugar on the molecule and make something like a little Trojan horse that can get inside the cell really easily. Okay, so just to wrap this up, this is what the drug looks like. We modeled this. Here's some DNA. Here's the drug right here. There's the sugar I popped on. And there's the warhead. And the warhead still stays right in that DNA cleft. And it should still be able to work well. And it looks like that sugar is not going to keep the drug from doing what it needs to do. Okay? That's pretty exciting to me. All right? So where we end up with this? Well, my boss at Madison, he left, he went to University of Kentucky. He's not really interested in this kind of a research. I really don't have research sophisticated enough here to do that. So that's kind of a dead end right now. There's no current plans for picking that up. Right now there are drugs on the market that have antibodies, so antibody drug conjugate, there's actually 2. So that technology is actually still very good. But what about colloquium? Okay? Now, interestingly enough, the fellow who was over this whole project, he had enough. He just said, you know what, I quit. He left. He actually went to teach high school and college, which is a very noble profession, I might add. But right after he left, the company says, hey, wait a minute, let's look a little bit further at this. Maybe it's not that the drug is bad, but how we're administering the drug needs to be better. So they found that if you gave lower dosages, so not as much drug at one time, but gave it more frequently, people in a clinical trial actually lived longer. So they lived about 12 months longer than if they had no treatment at all, okay? And also, if you combine the colloquium, the Mylotarg, so it's on an antibody, with other anti-cancer drugs, people actually lived longer, 8 months longer, combining the colloquium with other drugs than just the other drugs by themselves. So what we think is it's not just the drug you get, but how you get it's important. And all of this comes because of FDA, their willingness to say, something's not working, we got to pull it from the market. So coincidentally, about 2 weeks ago, and I didn't even know this before I was writing this talk, they actually approved Mylotarg to be put back on market. And that, I think, is a really nice ending to the story. So, this is kind of where we were at the beginning of our talk, right? Pushing the rock up the hill. You're going to have this in your career at some point. This is going to be you. I don't want this to be you, okay? This is not who the FDA is. The FDA, you guys get the reference? Yeah. Right, okay, right? This is not the role of the FDA. This is really the role of the FDA right here, okay? And that's how sometimes you feel at the end of a long day or a project. But you get there by your knowledge, your tenacity, and in my field of work, the partnership with the FDA is really what helps these drugs come to market. Okay? Sorry we ran a little long, but I would like to take your questions if you would like to stay for a few minutes, and if there's anything I can help you with, please let me know.